Hello everyone and welcome to this lesson on adaptive immunity, T cells. As you may know, our surrounding environment is full of organisms that may potentially be harmful for us. As such, we have some external defense mechanisms that act as a first line of defense to protect us. If, however, there is a breach in those external barriers, then our internal immunity will come into play. Our body's internal immunity can be classified into innate or nonspecific immunity and adaptive or specific immunity. This classification is based on how our immune cells will respond to an invader. The innate immunity mainly involves cells like neutrophils and macrophages, which act to phagocytose the pathogen in order to get rid of it. Adaptive immunity, on the other hand, is mainly involving the lymphocytes, and it can be further classified into humoral immunity and cell-mediated immunity. The humoral immunity mainly involves B cells, which can eventually transform into plasma cells and release antibodies. It's these antibodies that will then go into the blood and tissue fluid and exert the immune effect. We'll discuss this in more detail in our lesson on B cells. In this lesson, we'll be focusing on the T cells, which are the main cell type involved in cell-mediated immunity. In general, our body is pretty good at dealing with most invaders that get past our external defenses using just the innate immunity and the humoral immunity. But there are some pathogens that are either strong enough to overcome them or have sneaky ways of getting past them entirely. When this happens, these pathogens are then able to gain access to our cells and they can enter into them. HIV is an example of such a virus. Once a pathogen is inside a cell, it is beyond the reach of both the innate immunity and the humoral immunity, as these types of immunity cannot act within the organism's cells. This is where the cell-mediated immunity comes into play, and where the T cells take charge. This type of immunity is especially useful for viral or parasitic infections, because these types of infections tend to be missed by other parts of the immune system, because they can hide themselves within the host cells. Now, the T cells, just like the B cells, are also lymphocytes. As such, they, just like the B cells, also originate from the bone marrow. But unlike the B cells, which stay in the bone marrow to mature, the T cells will migrate to the thymus gland and then mature there. The thymus is a small gland that's present behind your sternum or your breastbone. You can remember that T cells mature in your thymus because thymus starts with a T and we're talking about T cells, while the B cells mature in your bone marrow because bone marrow starts with a B and we're talking about B cells. After the T cells matured in the thymus, they then move to different areas known as lymph nodes. The lymph nodes are small bean-shaped organs that are present in various locations all across your body. Now, these T cells, once they're mature, just like the B cells, also have cell surface receptors that are going to be unique and specific for only one particular antigen. However, unlike the B cells, which in many cases can bind directly to the antigen, the T cells cannot do that even if the antigen was right there in front of it. You see, the T cells need antigens to be presented to them by the organism's own cells in order for them to bind to that antigen. The cells that present the antigens to the T cells are called antigen-presenting cells, or APCs. A very common example of cells that act as APCs is the macrophages. There are also other cells that can present the antigens to the T cells, including non-immune cells, but perhaps the most important example for us to know is the macrophage. Now, as we mentioned, similar to the B cells, the T cells also originate in the bone marrow. Here, you can see that these immature T cells have a large centrally placed nucleus with a thin layer of cytoplasm around it. But as you can see, since they are immature, at this point they don't have any specific surface receptors. From the bone marrow, these immature T cells will then migrate to the thymus, where they will mature. These cells will synthesize many different surface proteins, which will get exocytosed and attach themselves to the membrane to form the surface receptors. Once the T cells have their unique cell surface receptors, they're then known as mature T cells. These cell surface receptors are going to be unique from cell to cell, so different mature T cells will have different cell surface receptors. As you can see in this diagram, the receptor on this cell is different from the receptor on this cell, and so on. Each of these receptors will be specific for only one type of antigen, so all of these different mature T cells will bind to different specific antigens. You see, it's the shape of the receptor that determines which antigen it's going to be able to bind to. And since this shape is unique on each of these cells, they will all bind to different specific antigens. Only if the shape of the antigen is complementary to the shape of the receptor will they be able to bind to each other. Now, once the T cells are mature, they're then going to migrate to the lymph nodes. Once in the lymph node, each of these mature T cells will make a few copies of themselves.
Quick pause. If you're finding value in this explanation, there's so much more to discover at Alt Academy, where we break down every A-level concept with the same clarity. Now back to the video lesson.
As we know, the copies will be identical to the original cell. So now we have small groups of cells, and within each group of cells, each cell will have the same cell surface receptors. This kind of group of cells, where all the cells have the same receptor on their surface that is specific for the same particular type of antigen, is known as a clone. It's a fitting name considering that they're all identical copies of each other. So, for example, this would be one clone, and this would be another clone. Now, when one of our own cells, such as a macrophage for example, presents the antigen to the T cells, the T cell from the clone that has the complementary cell surface receptor to that antigen will be able to bind to it and become activated. This process of matching the antigen with the correct clone of T cells that have the complementary receptor is called clone selection, because you're selecting the correct clone from all the options that we have that can bind to this particular antigen. Once the T cell from the correct clone has bound to the antigen, it is triggered to start dividing by the process of mitosis. This is called clone expansion. So first you have clone selection, and then you have clone expansion, where the cells divide. As it continues to divide, some of those copies become memory T cells that will help with the next time the body encounters the same type of antigen, while the rest just become more T cells. This is in contrast to the B cells, which, when they divide in this way, form memory cells and plasma cells, the latter of which secrete antibodies. The T cells don't form any plasma cells because they don't secrete antibodies. After we've gone through clone selection and clone expansion, now there's two possible things that can happen depending on what type of T cell was activated. You see, T cells actually come in two different flavors. You have your helper T cells and you have your killer T cells. Let's first talk about what happens if helper T cells are activated. So, when a helper T cell is activated, it releases chemicals called cytokines into the bloodstream. Cytokines are chemicals that may be released by certain cells that will help to increase the body's immune response. When the T helper cells release their cytokines, they can activate a number of other immune cells. They can activate B cells to divide and turn into plasma cells, which will then produce antibodies to fight the infection. They can activate macrophages to carry out phagocytosis more vigorously, thereby getting rid of invading pathogens, and they can even activate killer T cells. On the other hand, when a killer T cell is activated in this way and undergoes clone selection and clone expansion, it will instead go around searching for cells that have been invaded by the pathogens. When our body's cells have been invaded by the pathogens, oftentimes they will display some antigens from that pathogen on their own surface as a sort of help signal to help the killer T cells identify them as infected cells. So the T cells are using these antigens that are being displayed on the surface of infected cells to recognize them as being infected cells. The killer T cells will then bind to these antigens using the receptor on their surface, and once they've bound, they will synthesize toxic substances that will be secreted onto those cells. This will end up killing both the infected cell and the pathogens that are growing and replicating inside it. Now, when our body's cells present the antigen to killer T cells, they do so by attaching it to a protein receptor on their surface called MHC1. MHC stands for the major histocompatibility complex. These MHC are protein molecules that our cells have that they can use to present antigens to our immune system. And our immune system can then use these antigens to identify foreign invaders. Now, there are two different types of MHC receptors, and each of these will only be able to present the antigen to a particular type of T cell. So, killer T cells will only recognize antigens that are being presented to it on MHC1 receptors. In contrast to this, helper T cells, which if you remember were presented antigens by antigen-presenting cells, need to be presented these antigens on MHC2 receptors. Now, see, the reason this is important is that the MHC1 receptors are present on nearly all of our body's nucleated cells, meaning cells that have a nucleus, while the MHC2 receptors are specifically mainly only present on the antigen-presenting cells. Since MHC1 is present on almost all nucleated cells, that means that any of these cells that get infected by the pathogen will be able to alert the killer T cells to that fact, and the killer T cells can then come and kill them and the pathogen. This is especially helpful for those type of pathogens that like to hide inside of our cells so they can avoid other types of immunity. But this way, the killer T cells can still get to them and destroy them. So the killer T cells help us to deal with those pathogens that have already invaded the cells. Okay, now let's summarize what we've covered. T cells are part of the adaptive immune response, and there's two types of T cells: your helper T cells and your killer T cells. The role of helper T cells is to enhance your body's immune response by helping to activate B cells, killer T cells, and phagocytes. The role of killer T cells is to kill infected cells, thereby killing the pathogens inside them as well. Both types of T cells are very specific and have receptors that are only going to be complementary to one particular type of antigen. And both types of T cells also need to be activated by antigen presentation before they can do their job. The helper T cells will only be activated by what are known as professional antigen-presenting cells, which are immune cells that are specialized for this very purpose, while the killer T cells can be activated by both professional APCs as well as other infected body cells that are presenting their antigens on their surface. Remember, we said that other infected body cells can also present pieces of the pathogen as antigens on their MHC1 molecules, and this can also activate killer T cells. Because they need to be activated before they can perform their function, this helps prevent the T cells from getting overwhelmed and inappropriately sending our immune system into overdrive. And with that, we've reached the end of this lesson. I hope you found it helpful, and I'll see you in the next one.
Hey there! If you found this A-level biology lesson helpful, why don't you head over to altacademy.org and sign up for our full A-level biology course. In addition to biology, you could sign up for physics, chemistry, math, or 4 other A-level subjects. Thousands of students have already taken the leap. What are you waiting for? See you there.